It's to bio now. It's gonna take some time, but we're gonna do it. This is day 43, continuation of 2 month syllabus completion challenge. I think I went too far ahead. Yes. From a single cell, you may wonder how a single cell doesn't goes on to form such a large organism. Growth and reproduction are characteristics of cells, indeed of all living organisms. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells. Each time they divide, these newly formed daughter cells can themselves grow and divide, giving rise to a new cell population. That is formed by the growth and division of single parent cell and its progeny. In other words, such cycles of growth and division allow a single cell to form a structure consisting of millions of cells. Alright, cell cycle. Cell division is a very important process in all living organisms. During the division of a cell, DNA replication and cell growth also takes place. Alright, all these processes, i.e., cell division, DNA replication, and cell growth, have to take place in a coordinated way to ensure correct division and formulated third, the formation of progeny cells containing intact genomes. Sequence of events by which a cell duplicates the genome, synthesizes the other constituents of the cell, and eventually divides into two daughter cells is termed as cell cycle. Okay. Also, cell growth in terms of cytoplasmic increase is a continuous process. DNA synthesis occurs only in what? Okay, this is going to be interesting now. Also, it is where the cell duplicates its genome, it synthesizes other constituents of the cell, synthesizes its divides and eventually divides itself to form more cells is known as a cell cycle. Although cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only during one specific stage of the cell cycle. The replicated chromosomes, DNA, are then distributed to daughter nuclei and by a complex series of events during cell divisions. These events are themselves are under genetic control. Yes, they are. They are under the genetic control now. In order to understand that, we need to understand the phases of the cell cycle. What are the exact phases that we need to understand of the cell cycle? A typical eukaryotic cell, yes. A eukaryotic cell cycle is illustrated by human cells in culture. These cells divide only once in approximately every 24 hours. However, this duration of cell cycle is basically varying from organism to organism and also from cell type. Alright, looks like nobody's live, but we are gonna do this. We are not gonna give up. Okay. This is not gonna be easy at all, but I know it's gonna take some time. And 5 minutes overall in. We can do it. This is gonna be a very interesting one. A typical eukaryotic cell. Okay. The duration of cell cycle varies from organism to organism and also from cell type to cell type. Yeast, for example, can progress through the cell cycle on only 90 minutes. The cell cycle is divided into two types, the interphase and the end phase. The end phase represents the phase when the actual cell division or the mitosis phase occurs. And the interphase represents the phase between the two successive AM phase. It is significant to note that in the 24 average duration of such cycle of human cell division appropriately lasts only for less than 5% of 24 hours. That is on average 1 hour of the time taken for cell division for a human cell, whereas the rest of the whole 23 hours is dependent is done through the interface, which is actually interesting to know because this is very interesting. And the end phase starts with the nuclear division corresponding to the what? Is corresponding to the separation of daughter chromosomes. Yes, it is separation to the daughter chromosome. Karyokinesis unusually ends with the division of cytoplasm. The interphase, though called as the resting phase, this is G naught. West. G naught. Oh shoot, what is this? Looks like it's happening again, guys. No recommendations. Alright. That's not good to hear, but I think it's still fine. Oh, this is not good. Mm-hmm. Ain't recommending anyone. Yo, can you hear me, guys? I don't know what's going on, but it's fine. It's fine, okay? You can do this. So, the M phase starts with the nuclear division corresponding to the separation of daughter chromosomes, karyokinesis, and usually ends with the division of cytoplasm, the cytokinesis. The interphase, we call the resting phase, is a time during which the cells are preparing for the division, or undergoing both cell growth and DNA replication in an orderly, unique manner. The interphase, G1 phase, gap 1 phase, S phase, synthesis, and G2 phase, the gap 2 phase. G1 phase corresponds to the interval between mitosis and initiation of the DNA replication, by the way. During G1 phase, the cell is metabolically active and continuously grows, but does not replicate its DNA. S or synthesis phase marks the period during which DNA synthesis or replication takes place. During this amount of DNA, the sperm cells doubles. If the initial amount of DNA is denoted as 2C, then it increases to 4C. Okay. G1 phase corresponds to the interval between mitosis and initiation of uh, DNA replication. During G1 cell becomes metabolically active, continuously grows, and does not replicate its DNA. But yeah, the initiation of the DNA replication and the cell growth is taking place in G1 phase. But in the S phase, the DNA replication or the DNA synthesis takes place. During this amount, the DNA cell doubles. If this amount of DNA is denoted as 2C, then it increases to 4C, meaning that it has doubled. Yes, it has doubled twice. 2C to 4C. And that's how we get the another method of writing it down. Okay, right. However, there is no increase in the chromosome number. What is that chromosome number? If the cell has diploid or 2n number of chromosomes achievement, even after S phase, the number of chromosomes remains the same. That is 2n. 2n is the thing here, okay? Let's hold it on this side because I hold enough. Because just hold it like this. Yeah. This is our whole bio textbook. You have to put this whole thing in our brain. This thing. Oh, shoot, that kind of jump scared me. Food you never get tired of. I don't really do, but that's a kind of good question, by the way. But still, nobody's watching my stream. It's fine. I mean, it's not gonna hurt at all, right? I always wonder, like, why is the population of reaching this? But it's fine. You cannot give up. Okay, cannot give up. See, so we know that DNA synthesis is taking place, and for that DNA synthesis, what we need to know is that 2C gets increased to 4C. That is, the DNA gets increased. However, there is no increase in the chromosome number. If the cell had diploid or two number of chromosomes, even after estrase, the number of chromosomes remains the same, that is 2n. In the animal cell, during the S phase, DNA replication begins in the nucleus, and then the centriole duplicates. Okay, DNA replication takes place in the nucleus of the cell. Where does it take place? In the nucleus of the cell. DNA replication takes place in the nucleus of the cell, and the centriole duplicates in the cytoplasm during the G2 phase. Proteins are synthesized in the preparation for mitosis, while cell growth continues. And synthesized in preparation for mitosis, while cell growth continues. Okay. Some cells in adult animals do not appear to exhibit division. For example, heart cells and many other cells divide only occasionally, as needed to cells that do not divide further in situ. Phase to enter an inactive stage called quiescent stage. Or what is quiescent stage or G0 stage? What do you mean by this of the cell cycle? Cells in this stage remain metabolically active, but no longer proliferate unless called on to do so, depending on the requirement of the organism. In animals, mitotic cell division is only seen in the diploid somatic cells. However, there are a few exceptions to this, where haploid cells by division also takes place. For example, male honeybees against this. The plants can show mitotic divisions in both haploid and diploid cells. From your observation of examples of alternation of generations in plants, chapter 3, identify plant species and the stages at which mitosis is seen in haploid cells. This is the most dramatic period of the cell cycle, that is the end phase. We need to know this one, involving a major recognition of virtually all components of the cell. Since the number of all chromosomes in the parent and progeny cells is the same, it is called as the equational division. Okay. Though for convenience, mitosis has been divided into four stages of nuclear division. Karyokinesis. It is very essential to understand that cell division is a very progressive process and very clear-cut lines cannot be drawn between various stages because the karyokinesis involves all following four stages. Prophase, which is the first stage of karyokinesis of mitosis, follows S and G2 phase of interphase. In the S and G2 phase, the new DNA molecules are not distinct but interlinked. Okay. Can do this. Oh, hey there, bro. Finally, I see someone. Oh, shoot, I thought there was nobody. Do you mind chatting in the stream? Because, dude, oh, shoot, he left. Oh, shoot. I finally saw someone, dude. Like, bro, there was one person who was watching me. Yo, I thought my stream went dead. I thought my YouTube algorithm recommendation wasn't working. It, now, there are two ways, okay, to understand this. Usually, whenever I check, after 3 to 4 minutes, the stream will pop up to many viewers. But this time it's like after 15 minutes, which gives me a confirmation that it could be any either of my friends that are watching my stream. Well, I have no idea because there's no one right now here, But yes, I get the idea of someone studying here. Hmm. So, anyways, oh, okay. Oh, hi guys. Oh, shoot. Oh, finally I see someone. Oh, wait, no, wait. YouTube recommendation algorithm is working. Yo, yo, how are you guys doing? See, this is my thumbs up. I'm studying biology. Hi, hola, it's Monsieur 67. How are you doing? Oh, hell. Ocean Kings. How are you, Ocean Kings? Guys, do you mind subscribing to my channel? Oh, shoot. Goodbye, goodbye, goodbye. Yo, hi. Oh, shoot. Hi, Basilisk 2.12. Oh, shoot. Bro, that is crazy.
शाम मेरे बेटे
Hmm? रुको रुको
क्यों क्या हुआ
पढ़ लो
पढ़ो जरा जरा साफ सुथरा
मैं
बोलो
आता हूँ मैं आता हूँ
ठीक है लेकिन जरा साफ सुथरा में बोलो बेटा
मैं आता हूँ मैं पढ़ ले का आता हूँ
ओके जरा
कुछ से पढ़ ले कुछ
नहीं मेरी बात सुनो किससे बात कर रहा है
किससे भी नहीं वो पढ़ता हूँ सच सच लाइफ में करते
ठीक
रहते
है मैंने देखा था प्राइवेसी में मैं इंटरफेयर नहीं होता लेकिन जरा अच्छा मुंह हाथ धो को जरा क्लीन रहो बेटा
ओके
प्लीज
मिया
क्यों
ओके
क्यों क्या हुआ
यहाँ पे नहीं देख मैं पढ़ता हूँ मैं जाता हूँ मैं खुद रहता
बढ़िया बेटा
What your name? My name is Totality Hayo. My name is Totality Hayo, guys. Oh shoot, man. Oh my god. Finally, YouTube recommendation algorithm has worked. How you guys doing, man? Hi. I'm studying biology right now, if you don't know. Oh shoot. I never thought these many people come with this kind of time. My name is Totality Hayo. I won't say my real name right now. Cool. I see. Hi, Mia. How are you doing? Okay, you know what? How are you guys doing? Oh, shoot, man. Oh, okay. Finally, I see someone. I thought my stream went dead for a second. Not for a second, for 15 minutes straight. Then these people popped up. Oh, thank you. Thank you, YouTube. Thank you, God. Prophase, which is the first stage of cryokinesis of mitosis, follows the SNG2 phases. Yes, it is hard. It is hard. मैं आता हूँ मैं मुंह धो ले कुछ आता हूँ Yes, it is really hard, dude. I swear it is hard. I always try my best to work hard. I stream consistently. Yeah, it is hard, man. How is your days going? How are you guys doing? How are you guys doing, man? Wow. Many people, okay, many people are going down, but it's fine. Good, good. Hi, MD Muhabbat Hussein. How are you doing? Oh, I'm doing great. I'm fine. I'm fine. Alhamdulillah. I'm fine, brother. Alhamdulillah, fine. I'm great. All right. That was great. But let's study. I cannot just So, prophase. In prophase, there is one thing we need to understand. Okay, I'm so sorry. This will look boring, but trust me, I try my best. I always not give up. I always move on forward. We can do this. Okay, if you want, you can chat and talk with others. I'll keep like responding, but right now we'll be studying at the same time. Whew. Okay, this is gonna be not easy. Prophase, which is the first stage of karyokinesis of mitosis, follows the S and G2 phases of interphase. In the S and G2 phase, the new DNA molecules are not distinct but intertwined. Okay, cool. Yes, I know. Hi, Skyman. How are you doing? Oh man, oh I'm so happy. Oh, I'm so happy, man. Like literally, I had no viewers watching me that time. By seeing these many viewers, makes me happy that YouTube recommendation has popped up. It was not going up for straight so long, man. Oh, I thought this was gonna be really bad. Oh, thank you for the hearts. Thank you for the hearts, man. Oh, thank you. That looks like a really huge of support. Wow. Shoot. Oh, man. Only there are two people watching, but I'm still happy because, see, show me your deodorant. Um, my deodorant. Wait a minute. I don't know. Maybe 1 hour or more than that. It's night though. Like it's gonna hit 11 o'clock soon, but not right now. Show me your deodorant. Okay, Cisco. Okay, you're gonna do that. Just wait. Yeah, this is kind of deodorant. This is not actually a deodorant, but yeah, we kind of use this as a deodorant. Sorry for the dot dots. I went away for a while. This is a very weird question though. Cool, I know. I don't know how is that cool though, but yeah. Oh shoot, let's study now. Prophase, which is the first stage of the karyokinesis of mitosis, follows S and G2 phase of interphases. In the S and G2 phases, the new DNA molecules formed are not distinct but intertwined. Good enough. Okay, thanks, dude, Cisco. Shout out to Cisco, Mia, Skyman, MD Muhabbat Hussein, and shout out to all. And man, okay, okay, I get back. Okay. Oh, thank you for the hearts. Oh, thank you. Oh man. Okay, the S and G2 phase. Prophase is marked by the initiation of the condensation of chromosomal material. By this, the chromosomal material becomes untangled during the process of chromatin condensation. The centrosome, which had undergone duplication during the S phase of interphase, that is now begins to move towards the opposite poles of the cells. The completion of prophase can thus be marked by the following characteristic events. Chromosomal material condenses to form compact mitotic chromosomes. These chromosomes are seen to be composed of two chromatids attached together at the chromatids. Mm, the centromere, sorry, my bad. Okay, centrosome which had undergone duplication uh, during interphase. Hi, you're back. Nice to meet you again. Oh, you know what? Gonna subscribe to your channels. Just wait for a minute, okay? I'm gonna subscribe to your channels. I'm 16. I'm old enough to live stream. Yeah, I'm gonna subscribe to your guys. Since you're watching my channel, man. Like, dude, I appreciate each and every viewer. Love you all, man. Let's check. Okay. Let's go to your. Wait a minute, okay? Just wait a minute. Let's go. Alright. We're going to mine and Here we go. We can check this out. So, all right. We're gonna do this. Just wait a minute, okay? Just wait for a moment. Oh, hi. Hi, Hisham Abdul Rahman. How are you doing? Okay. Just wait a minute, okay? Okay. So, um, okay. I subscribed. Mia. Thank you, MD Muhabbat Hussein. Here you go, Muhabbat Hussein. You're gonna get subscribed too. Um, Now you have two subscribers, Muhabbat. Where is Skyman? Skyman. Skyman. Go to your channel and subscribe. I'm your first subscriber, Skyman. Okay. Cisco. Yes, Cisco too, huh? Cisco, go to your channel. And I'm your 11th subscriber. No, I'm not a Roblox player. I know what's the game called, but I'm not a Roblox player. When I was young, like at least like 11, 12, or 13, I used to be a Roblox player. I used to play Minecraft. But yeah, I know that game. It's a really good game though. Um, my sister watches it. Like others play. Minecraft, yeah, I used to play Minecraft. I used to play Minecraft. When I was small, I used to play a lot of Minecraft. It was one of my favorite games. Anyways, metaphase cells at the end of prophase. I'm from Middle East, but I don't speak Arabic though. I just don't speak Arabic. I see. Okay. I do not play any games, guys. I just study, right now. But yeah, if you talk about any game, I might know that game. Because I have played it previously, but there are some games which I've never played. I kind of speak Urdu, if you don't know. Actually, I'm an Indian, if you don't know. Do you play sports? Yes, I play only some sports. Sometimes I would play football, but yeah, not much. I'm not a big football fan or a cricket fan, or I don't play much of badminton or any other kind of sport. Yeah, but I used to play football. Okay. Cells at the end of prophase, when viewed under the microscope, do not show Golgi complexes, endoplasmic reticulum, nucleolus, and the nuclear envelope. Yes, these get condensed to form the chromosomes. Yeah, it's fine, Mia. It's fine. I understand because you're not above that age restriction, but you can still do it. Try making some videos so that you can become more popular. I know you can do it. Just wait for that age limit to be crossed, okay? Follow the YouTube guidelines and you will do it, okay? You can do this. Yes, you're welcome. You can do this. Now, metaphase. I'm just studying biology if you don't know. Complete disintegration of the nuclear envelope marks the start of the second phase of the mitosis. The second phase of the mitosis. Which part? Karnataka. Karnataka. You know that? Southern Indian state? Complete disintegration. I'm a Southern Indian, but yes, I can speak Urdu too. Complete disintegration of the nuclear envelope. What is the nuclear envelope? It marks the start of the second phase of the mitosis. Hence, the cytoplasms are spread through the cytoplasm of the cell. By this stage, the condensation of chromosome is completed and they can be observed clearly under the microscope. Yes, sir. Yes, sir, guy. Yes, sir, Skyman. Yes, sir. Which are you from India too? And which part? I don't know. Um, I don't know which part of India you live in, but if you want to mention, it's fine. You can. Okay. Mm-hmm. The cytoplasm, and here condensation of chromosome is completed and they can be observed clearly. Kerala, I see. Southern Indian here. I'm from Bangladesh. Oh, wow, that's great. Muhabbat Hussein, I see. So you're nice. You're from Bangladesh. What about Mia? Where are you from, Mia? Do you mind checking that out? Okay, wait. You know what? Let me just check. I see him. Oh, okay. That is crazy. Many people subscribe to my channel. Oh, shoot. Oh, shoot. Yo, that's crazy. That's crazy. Thank you for subscribing to my channel too. Wow. Thank you. Thank you. Thank you. Thank you very much. This means that we are gonna reach 70 subscribers now. Only so close to 70 subs. We can do this, guys. I get back soon because my horses is cold, and so I got to take them. And so I get back in 20 minutes. Okay? Yes. Take your time. Take your time, Mia. Take your time. Right now, I think I have 69 subscribers. According to me, I have 69 subscribers. Only one sub left for 70 subscribers. It's fine. I'm still doing it. Yes. Take your time, Mia. You can go if you want to. You can come back whenever you want. Um, I'm not sure whether I'll be live at that time, but yes, I'll try my best to keep on going. What is your dream? My dream is not that specific, but when I grow up, I want to go into engineering. Now, you might be feeling weird because I'm studying biology here, but then why am I going for engineering? My stream is actually both mathematics and biology. I love biology and maths at the same time. I chose both of them. So, I want to go for engineering. I want to learn more software. I want to create some apps and help the world throughout it. So, yeah, my ambition is to create some apps and even start up a YouTube channel. That was the dream, and yeah, I did. I can still do it though. Yes, we can still move on now. Oh, we cannot give up. Whew. Oh, sure, okay. This then is the stage at which morphology of chromosomes is most easily studied. At this stage, metaphase chromosome is made up of two sister chromatids. Uh, yes, which are held together. Okay, they are held together by the centromere, a small disc-shaped structure at the surface of the centromeres called kinetophores. These structures serve as a site of attachment uh, of spindle fibers formed by the spindle fibers to the chromosomes. And okay, oh shoot, oh shoot, oh shoot. Oh, this is not a good situation. Oh, okay, now let's do it. That wasn't good at all. I was gonna get stuck. I was gonna get stuck so bad. Oh shoot. Oh shoot. I don't know. I just don't wanna disclose it. Okay, okay. Cannot give up. We cannot give up. We still can do it. These structures serve as the sites of attachment of spindle fibers formed by the spindle fibers to the chromosomes and that are moved into position at the center of the cell. Hence, the metaphase is characterized by all the chromosomes coming to lie. Hmm, okay, coming to lie. मैं सोने जाता हूँ और oh, this is great. I should. This is not great. Okay, metaphasic plate, and then spindle fibers attached to kinetophores of chromosomes is known as the metaphase plate, and that is how we understand that stuff. Chromosomes are moved to spindle equator and get aligned across the metaphase plate through spindle fibers to both poles. Anaphase. At the one set of anaphase, each chromosomes are arranged at the metaphase plate is split simultaneously. The two daughter chromatids, now referred to as daughter chromosomes of the future daughter nuclei, begin their migration. मालूम begin their migration towards the two opposite poles. As each chromosome moves away from the equatorial plate, the centromere of each chromosome remains directed towards the pole and hence at the leading edge with the arms of chromosome trailing behind. Thus, anaphase stage is characterized by the following key events. Centromere splits and the chromatids separate. Chromatids move towards the opposite poles. Okay, let's go through this. Okay, we can do this. Oh shoot, you're doing it, guys. You're doing it. I believe in you. Thank you very much. Thank you very much, Muhabbat Hussain. Thank you very much for believing in me. Thank you very much. So they lose their identities and they form the chromatin material strings to connect each other to the two poles. This is the stage which shows the following key events. Chromosome clusters of opposite spindle poles and their identity is lost as discrete elements. Nuclear envelope is developed around the new chromosomes clusters at each pole, forming two daughter nuclei. Then nucleolus Golgi complex and ER reform, that is the endoplasmic reticulum or reform. Yeah. Cytokinesis. Now this is very interesting. We need to understand cytokinesis, then the significance of mitosis. Let's do it. Sorry for making this content very boring, but trust me, I try my best every time. I'm not gonna give up. Okay? You're not gonna give up. Mitosis accomplishes not only the segregation of duplicated chromosomes, the segregation of duplicated chromosomes into daughter nuclei, karyokinesis. But the cell itself is divided into two daughter cells by the separation of cytoplasm called the cytokinesis. Okay, at the end of which cell division gets completed. In an animal cell, this is achieved by the appearance of furrow in the plasma membrane. The furrow gradually deepens and ultimately joins the center, dividing the cell cytoplasm into two. The plant cells, however, are enclosed by relatively inextensible cell walls, so making furrows is not actually a really interesting method of actually doing this. So, yes, we need to have that idea basis. Yes, 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 we need to have that idea basis. Okay, okay. Oh, phew. Okay, all right. We cannot give up. Sorry, the equation. No. My bad. Mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei. Yes, we can do this. We can do this. We can do this. Mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei, karyokinesis, but karyokinasis, my bad. I don't know why my pronunciation is going so bad. But the cell itself is divided into two daughter cells by the separation of cytoplasm called cytokinesis, at the end of which cell division gets completed. Okay, in an animal cell, this is achieved by the appearance of a furrow. Yeah, you can go, man. It's fine. You can go. Bye. Thank you for subscribing, dude. I'm really happy that I actually helped some people. Thank you very much. Bye, dude. You can go. Bye. Oh, sure. Okay, you can go now. Ha- I will study. I'll have to keep on. I have to continue, I cannot give up. I can do this. So you can do this too. Bye. Okay. Therefore, they undergo cytokinesis, but by different mechanism. In plant cells, cell wall formation starts, and this in the center of the cell grows outward to meet the existing lateral walls. The formation of new cell wall begins with the formation of a simple precursor called the cell plate. Yes, a cell plate or a simple precursor is formed, and that's how we get the main different idea or symbolic pathway here that, oh, this is actually working. Oh, this is a great method. Yes, it is a great method. Wait a minute, is my screen getting heated off? No, it is not getting heated off. It's fine. Mitosis significance. Okay, with syncytium, reductional division. It is the equational division, or it is usually restricted in the diploid cells only. However, okay, wait a minute, I have to read through this again. Mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei, but the cell itself is divided into two daughter cells by the separation of cytoplasm called the cytokinesis. At the end of which cell division completes in an animal cell, this is achieved by the appearance of a furrow in the cell membrane. Alright, okay. In the plasma membrane, the furrow gradually deepens. Yes, it deepens here and wink. Yes, it deepens and it eventually comes in contact with the center of the cell, and therefore the cell cytoplasm gets divided into two, and therefore we see this division. Now, this is not the similar case for plant cells because plant cells have something called cell wall, which is inextensible and rigid. So making furrows is not a method here. Instead, there is a new cell wall formed that is a simple precursor and it is lateral to these cell walls, which is known as the cell plate. Now, this cell plate is actually representing the middle lamella between the walls of two adjacent cells. Yes, it is basically representing the middle lamella, and therefore, once this is done, what exactly happens is that once the middle lamella between the two walls of the two adjacent cells, the cytoplasmic divisions, organelles like mitochondria, plasmid plastids get distributed between these two plant cells. Once they are these constituents will get divided, they will actually make more of these, and since they make more of these, what we need to get the idea is that since there are more of these made, one will go into the one cell and the other will go to this cell, making two in one of these. And sometimes there is are chances where we see multinucleated cells, and these multinucleated cells are basically done when karyokinesis takes place, but not the cytokinesis. And this is known as syncytium, my bad, syncytium. What is it? Syncytium. And this is basically found in the liquid endosperm in coconut. Yes, the coconut has liquid endosperm, and that liquid endosperm is basically the idea of the formation of syncytium. Now, the significance of the mitosis. Yes, we need to have the okay, man, it's very fine. Wow, it's fairly fine. Wait a minute. Here we have this done. Oh shoot. Okay. Whoa, 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 whoa. What just happened here? That was random. I see. I see. That was so, so, so random. I see. Mm-hmm. That is so randomized. Am I shaking that much? Also, my voice. That is so randomized. Am I shaking that much? Also, my voice. Yeah, yeah, yeah. My voice is really cracked up. Also, my voice. Yeah, yeah, yeah. My voice is really cracked up. Okay. Mm-mm. It says upcoming challenge, huh? Yes, it says upcoming challenge. It really says that. It really says it. Anyways, what am I doing? You shouldn't be doing this, huh? We shouldn't be doing it. We cannot be doing these kinds of funny tick 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 stuff, right? Yes, we should be doing that. Let's concentrate. Mitosis or the equational division is usually restricted to diploid cells only. However, in some lower plants and in some social insects, like the honeybees, that is the male honeybees, haploid division haploid cells also divide by mitosis. Yes, so uh, Oh, that's the thing here. However, in similar plants and some, okay, it is very essential to understand the significance of this division. Yes, we need to understand the significance of this division in the life of an organism. Are aware of some examples where you have studied about haploid and diploid insects. Now, mitosis usually results in the you know, production of diploid daughter cells with identical genetical components. Now, the growth of a multicellular organism is due to mitosis. The cell growth results in disturbing the ratio of the nucleocytoplasmic ratio. The cell it also helps in the growth of a multicellular organism. A very significant contribution of mitosis is cell repair. Yeah, that's very important to know. The cells of the upper layer, the epidermis, cells of the lining of the gut, and blood cells are being constantly replaced. Okay. Mitotic divisions in the meristematic tissues, the apical and the cambium, that is the lateral meristem, which we are going to study in plant development and growth, result in the continuous growth of plants throughout life. That is the 13th chapter of our biology. Nice. Now, meiosis. Now, what is meiosis basically? What exactly meiosis explains us? Can you elaborate it for us? Yes, we'll do it. Okay. Now, production of offspring by sexual reproduction. Okay, I shouldn't have said that here, but I should be doing this because I'm studying meiosis. Includes the fusion of two gametes, that is the male and female gamete. Yep, yep. Hi, hi there, hi there, Weaver. How are you doing? Oh, he left, he or she have left. I don't know who was that, but yeah, he or she has left. I don't know who was that random Weaver, but let's see. Okay, it was so randomized. Alright, we are gonna do this. Each with a complete haploid set of chromosomes. Now, gametes are formed from specialized diploid cells. This specialized kind of cells division that reduces the chromosome number by half results in the production of haploid daughter cell. This kind of division is called the meiosis. Meiosis ensures the production of haploid phase in the life cycle of these reproducing organisms that require mating is what whereas yeah whereas a fertilization restores this from haploid to diploid phase. We come across meiosis during gametogenesis, that is gametogenesis meaning the formation of the gametes in plants and animals. This leads to the formation of haploid gametes. Yes, the key features of meiosis are as follows. Meiosis involves two sequential cycles, meiosis 1 and meiosis 2, but only a single cycle of DNA replication takes place. Yes, only a single cycle of the DNA replication is taking place here, and yeah, that's much here. But the meiosis one is initiated after the parental chromosome have replicated to produce identical sister chromosomes. Okay, wait. Initiated after the parental chromosomes are replicated to produce identical sister chromosomes at the S phase. Okay. Meiosis involves pairing of homologous chromosomes and recombination between non-sister chromatids of homologous chromosomes. Alright. Four haploid cells are formed at the end of meiosis too. Hmm. Meiotic events can be grouped under the following phases. Whew. Prophase 1. The prophase of the first meiotic division is typically longer and more complex when compared to the prophase of mitosis. It has been further subdivided into the following five phases based on chromosomal behavior: leptotene, zygotene, pachytene, diplotene, and dikinesis. During the leptotene, the stage. Oh, hi there, Fever. Hi, how are you doing? This is me, Totality Hayo, and I'm very happy to see you. How are you doing? Oh, he left. It's fine. Let's continue. Okay, that is the very weird stuff. Oh, that was very weird. That's very weird. Okay, that was so utterly weird. I have no idea how to express that up. Oh shoot, that was so weird, man. Okay. Yep. That's weird. That's weird. How should? Anyways, the chromosome. During the leptoteen stage, the chromosome becomes gradually visible under the light microscope. The compaction of the chromosome becomes continuous through the what we can say here is very interestingly knowing as something. Okay, so what exactly are we talking about here? That the leptoteen is a basically a stage where the chromosome is basically gradually visible under the light microscope, okay? And the compaction of the chromosome, that the chromosomes actually get compact, continues throughout the leptoteen, okay? That is there, fine, it's fine, we got this. And what else? What else do we need to know? What else do we need to know? Hmm. Yeah, they start pairing together. No, wait, what? The compaction of chromosomes becomes continuous throughout this. Okay. Okay, got it now. Starts pairing together, and this process is called synapsis. Such paired chromosomes are called homologous chromosomes. Okay, all right, I see. Electron micrographs of this stage indicate that chromosome synapsis is accompanied by the formation of complex, that is known as synaptomial. Oh my God, synaptonymal complex. The complex formed by a pair of synapsed homologous chromosome is called a bivalent, all right, or a tetrad. However, prophase one are they are more visible at the next stage, and the first two stages of prophase one are relatively short. Yes, they are relatively short and lived compared to the next stage, that is Pac-18. And what happens is that during this stage, the four chromatids are of each bivalent. Okay, okay, all right. Four chromatids of each bivalent chromosomes becomes distinct and clearly appears. I'm so sorry. Oh, hi. Hi, Mia. How are you doing? It's fine. It's fine. How are you doing now? Nice to see you again. I'm still here. Don't worry. I'm still here. It's fine. And also, golf for Bund. Okay. Well, sorry. It's fine. It's fine. It's fine, Mia. Where do you live, by the way? I'm kind of curious. I meant good. Oh, okay. I see. It's fine. How are you doing? And I just had a doubt. Where do you live? Like, I asked some people, they said some of them live in Bangladesh and some other people live in India. I was like feeling, what about you? Where do you live? If you don't know where I live in Middle East. I'm a Middle East person, but I'm originally an Indian, if you don't know. So yes. And how are you doing, Mia? Thank you for okay. How are you doing? And thank you for subscribing to my channel. Thank you very much. Thank you. Good. Yes, it is good. Thank you, Mia. Um, is Mia your real name or it's like your surname? Like I'm confused. Mia is your real name or a surname? I have a doubt. That's why I'm just asking you. Oh, okay. Yes. Okay, nice. I can see the emojis. Emojis. Yes, I can see those emojis. Nice. So you play Roblox, I guess. Like from your channel, I can see that you're a Roblox player. I see. What is your favorite Roblox game? can you like give me some of your favorite Roblox games? I want to know that because I just want to hear from it. Like any Roblox game that you like you described 99 Nights in the Forest, so that's your favorite game? That's your favorite game? Oh, nice. I see. Nice, nice. That's great. I see. Grey Garden. I see. Nice. Oh, okay. Grey Garden. Nice. I see. Yeah, nice. It's fine. It's fine. That's really. I know those games. Those are really good games. Nice, nice. Okay, yeah. How are you doing, Mia, right now? Good? Oh, okay, okay, my bad. I said so many times. I'm so sorry. Oh, should. Okay. How's life right now? Is it tough for you? Is it good? Is it like fine? And where do you live, by the way? I just want to know because you feel like you're from Europe, or I don't know. I'm not sure whether you're from Europe or not. Uh, can you just give me your place? Norway. Oh, wow. Nice country. I see. Is it cold right there? Like, is it very cold right there in that country? I'm not sure. I think the temperature is very low right there. Yeah, really cold. I knew it. I mean, yeah, it's of course winter. Here in Middle East, it's not that cold. It's like temperate. I think it's somewhere around 27 to 28 degrees Celsius. So it's not that cold. I understand though. So stay safe. Okay, you're doing great. All right. This is kind of very interesting to know, but you're one of my viewers who's still standing up to here and watching me. It says that you want to also become like a streamer, like me, and want to stream. I understand you can do it, but for now you gotta obey the YouTube guidelines and you gotta do the YouTube videos. So if you start continuing with your YouTube videos journey, you might attract more viewers, and those more viewers will start watching you in your live stream. So you can still make videos, okay? You still can do it. Don't worry. I know you can. It's a little bit hard. Yeah, I understand. I understand. Don't worry. Don't worry, Mia. I still understand. And remember, since I subscribe to your channel, you subscribe to my channel, you can give me a fist bump. See? I gave you a fist bump. Nice. Okay, fist bump. See? Fist bump. Let's go. Like, we actually like, yeah, yeah, I know. You did it too. You subscribed to my channel, I subscribed to yours. Yes. Right now, what's the time right there? Is it like morning or is it like afternoon? I think it's afternoon. I'm not sure. I'm just asking. Could be afternoon right there. Night. Oh, I see. Anyways, Mia, looks like you can go if you want to. If you wanna go, you can. If you wanna stay, you can stay. Depends upon you. I'm still gonna continue a bit of studying. I think I'm gonna end my stream soon, after 5 to 10 minutes. But yes, it was nice meeting you. If you want, you can still stay. Depends upon you. All right. I'm not gonna force you at all. You're my viewer, after all. And yes, it was actually nice me seeing you, meeting you. Yes, yes, yes. You can have a nice sleep, okay? Good night. Have a nice sleep. I love you. Bye. Yes. Bye, bye. See you later. Yes, I'm gonna go study now. I will end my stream a bit later. So yes, after 5 to 10 minutes, I gotta go. Bye. Have some fun. Bye. Now I gotta continue. I will lock in now. So what I know is that the prophets are relatively short-lived compared to the next stage. That is, prophets which are relatively short-lived compared to the next stage. That is the Pakitian stage. Now, during the Pakitian stage, during this stage, four chromatids are basically, are each bivalent chromosome becomes distinct and clearly appears as tetrads. I eat treat my viewer. I love all of my viewers. Thank you, viewers. You're doing it. Oh, let's do it. The complex formed by a pair of synapsed homologous chromosomes called tetrads. Okay, two homologous chromosomes. It feels like it's time to end the stream at the same time, but let's just wait, okay? I just called us diets of cells. Oh, phone is being heated up. We can do this. We can do this. Tetrads of cells. We read the whole one chapter now. So I think it's time to end the stream. Um, it was a pretty good run. Let's see. My voice is also cracked down a bit. I don't know what's going on. Like I want to hear it. All right. Nothing much here. Hmm. All right. Let's go. All right. Bye, guys. Gotta go now.